This is a continuation of your cell communication notes, and we are going to look at the nervous system actually to put some of this in more context. So this is the big sheet that we're going to go ahead and use, and we're actually going to start down here on this side with this little drawing kind of in the bottom middle. Come down here, zoom in. All right, that's good. So this is a typical nerve cell. On one end of the cell, you have the body of the cell with all of these little dendrites. So this is the body with all the cytoplasm. The nucleus is here, and all of these little extensions that are kind of sticking out are called dendrites. And those are going to actually receive signals from adjacent neurons. And this long portion here is what's called the axon. That's what's going to send the signal down to the end of the cell. And these little boxes here are actually cells that help speed this signal up, this impulse, this electrical impulse up. It makes them go quicker. So these are called Schwann cells. You don't really have to know this, but I'm just going to go ahead and give you the extra info just for completion purposes. Schwann cells, oops, those should be two N's. Schwann cells, and they make up what is called the myelin sheath. The myelin sheath. Again, this is kind of a little extra info that, we'll talk about it. And basically this is insulation. This serves as insulation. Think of the axon as a wire. This is insulation that speeds up the message down the axon. And these are actually flat cells that wrap around the axon. These Schwann cells are kind of like flat pieces of paper, if you want to think of them like that wrap around the axon and speed this message up. And the message is always going to go from the body down through the axon in this general direction. It's not going to go backwards. It's a one direction movement here. Oops. If you have a neuron in the body, you're probably going to have these Schwann cells on that nerve cell because you have potentially feet and meters of distance to cover if you're sending a signal, possibly from the base of your foot up into the base of your spine or vice versa. So some of your neurons in your body are actually quite long, and they benefit from having these Schwann cells on them to help speed that signal up. But if you are in a brain, brain cells typically don't, or brain neurons or neurons in the brain typically don't have Schwann cells, and they don't really have myelin sheath. So down here is a neuron in your brain, and there is no myelin sheath on these cells. They transport the signal slower, but slower is also incredibly quick, like milliseconds is how long it takes the signal to propagate. So it's not that much slower, but comparatively it's a little bit slower because they don't have those insulating cells. So this would be slower, and this would be like in your brain. It's also much shorter distances to travel in the brain. And if you think about all the work that this axon and the body of the cell is doing, it's going to require a lot of ATP. So these cells often have supporting cells called glial cells that basically just give the cell nourishment and support the function of the cells. And this is extra info. You don't have to know it. But think of these cells as supporting cells. Support cells. They nourish, oh, let's say nourish. They nourish and they help regulate. They help regulate, in particular, the extracellular fluid around the nerve cell. But that's what glial cells do. So that's just kind of a little introduction to the anatomy of a cell, of a nerve cell. So if you look up, zoom back out, what we have here is a nerve cell. It's not nice and thin like we have in this drawing towards the bottom. It's a larger, fatter nerve cell so we can see what's going on inside. But here are the dendrites on the left. I didn't really draw them a lot just because it takes up space, but this is where the body of the cell is. Here's the nucleus right here. Here's the axon, and then here's the end of the neuron. There's a space, and go ahead and label this, called the synapse. Go ahead and type it. The synapse, and that's a space in between the end of one neuron and the start of an adjacent neuron, and we'll talk more about that synapse in a second. So let's look at the anatomy of the neuron. What you're going to see, we're going to start over here on the left, is that we're going to start at the body with the dendrites. You have a ligand-gated ion channel. Remember, a ligand is just a molecule. It's a signaling molecule that's going to cause a cellular reaction, right? So we're going to end up starting here and moving to the right. And again, a ligand is just a molecule. And we'll talk about what molecule this is in a minute. It's some kind of molecule. And this ion channel is going to open in response to this ligand. And we'll see that this is actually a sodium ion channel. But it's going to respond to that ligand. And if you look along the axon, you're going to see all of these different channels and some pumps. And I've gone ahead and I've labeled them up here. You have voltage-gated ion channels in the axon. Notice that it's different than the ligand-gated channel in the body where the dendrites are, where the soma, the body of the cell is. These are voltage-gated, not ligand-gated. So these boxes that you see here, here's one, right? These are sodium ion channels. Remember, they're voltage-gated. They respond to changes in charge. These triangles are potassium, and these circles are calcium. And if you notice, the axon has a lot of the sodium and potassium channels, whereas the terminal region, the end region, is the region that has the calcium ion channels, and we'll see how that works in a minute. You're also going to notice that along the axon are protein pumps, sodium-potassium pumps, and this is going to require ATP. Remember, the channels are facilitated diffusion. This is no energy needed. But the pumps, the sodium-potassium pumps, do require ATP. This is active transport. This is active transport, and this was passive. Right. Before we keep going, I just want to make a note that these sodium channels are very, very quick. They are very quick and fast to respond. These potassium channels are much slower, and this is going to matter when we talk about the change in voltage inside the cell, right? So let's take a look inside the cell. So the inside of the cell, the axon, has, in terms of positively charged ions, it has a lot of potassium. And that's really kind of it. Everything else inside of here is negative. It has negatively charged amino acids, chloride ions, and then this repeats, potassium again, chloride ion, potassium, amino acids. So the overarching composition of the inside of this cell is overwhelmingly negative. There are many, many more negative ions and negative amino acids inside the cell than there are positive potassiums, right? And if you're looking at just potassium and just sodium in particular, the inside of the cell has 150 micromoles of potassium compared to 15 micromoles of sodium, right? So the inside of the cell has a lot of potassium. Compare that to the outside of the cell, the extracellular fluid, which has basically the inverse. It has very, very little potassium and quite a lot of sodium. So when you think about the charges of a nerve cell, you can think of it as a salty banana, right? Salty because there's a lot of sodium on it, and a banana because, well, bananas are known for their potassium content. So it's kind of a little cute little trick to remember that the inside of the nerve cell, just like a banana, has a lot of potassium and it's salty. So the outside has a lot of sodium, right? The fact that the inside of this cell is negatively charged due to all of these negative ions This is why all of these little minus signs are just negative charges. These are all negative charges inside the cell. And the outside is positively charged compared to the inside, is what is called a polarized membrane, right? So this is the nerve cell at rest. This is the default for the nerve cell. The inside is negatively charged and the outside is positively charged. So we say that this is a polarized membrane, a polarized membrane. So actually, let's go ahead and draw this in this little graph down here in the corner. So at start, the inside, or the difference in the charges, actually, let's go back. The difference in the charges between the inside of the cell and the outside of the cell is actually negative 70 millivolts. This is what's called the membrane potential. There's a difference in charge on the inside and the outside of the membrane. And in a nerve cell in your body, this is about 70 millivolts. Negative 70 millivolts. That's the difference. For reference, if you're curious, an electrical outlet in your wall in the US has about a charge of 110 volts. If you're in Europe, since theirs is a little bit different, their outlets are different, they have about 220 volts. So your cells are negative 70 millivolts, so a lot smaller. So let's go ahead and graph this. So to start, right before anything happens, this is the nerve cell at rest, its default state. You have a resting potential of negative 70 millivolts, right? And because there's a difference in charge, we say that this membrane is polarized. We say that this membrane, is polarized. All right? So to see what happens next, we're just going to go ahead and we're gonna walk right on through this, and we're not going to stop. So you might have to pause if you need to take some extra time. But we're going to start on the left and we're going to move down through the right, and we're going to see exactly how this process works. It's going to take us a couple of minutes to go over it, but in real time, and I want you to note this, this entire process that goes from the left side to the right side down the axon only takes 1 to 2 milliseconds. This is how quickly it actually happens in your cells. 1 to 2 milliseconds, but even though it's going to take us some time to go through it. All right, so here we go. Ready, set, go. Here's the ligand. We'll talk about where it came from in a second. But that ligand is diffusing towards the body of this nerve cell. And because there are ligand-gated ion channels, when it binds to this ion channel, it's going to cause this ion channel to open. And I'm actually going to go ahead and change colors here. Because I'm going to try to keep this color-coded. And we've already talked to, oh, well, I'm just going to go back to blue. That's fine. Okay, I tried. Sorry, I tried. We've already gone over that this is a sodium ion channel. So when this opens, sodium, which is a positively charged ion that is in high concentration outside the cell, remember it's high concentration outside the cell, low concentration inside, is going to diffuse down its concentration gradient from a high to a low concentration into the cell. And because sodium is positively charged, that's going to cause the inside of the cell to start becoming positively charged as well. And this is where the signal starts to originate from. All of these negative charges, you can start drawing positive charges on them. This area of the cell becomes positively charged, right? Well, this sodium, and all these sodiums rushing into the cell, are going to diffuse down the axon. And as they diffuse down the axon, they progressively turn it more and more positive. So this positive charge spreads all the way down the axon, all the way down, right? It's like a wave, like in a football stadium, right? It propagates down the axon. Well, as it does that, it passes these sodium channels. And remember, these sodium channels, we're just going to look at sodium here first. These sodium channels are voltage gated. Well, hey, look, there's a change in voltage moving down the axon. And when that hits or passes these sodium channels, they too are going to open. And sodium, right, in a high concentration outside the cell, diffuses into the low concentration inside the cell. It's facilitated diffusion, and the process just continues. It continues and continues, and this happens all the way down the axon. The voltage change continues down. It activates more of these voltage-gated sodium channels, and more sodium diffuses into the cell. So eventually, as you make it all the way down to the end of the axon here, here's some more channels, more and more sodium is coming in, right? What happens is the inside of the cell, the axon of the cell, gets more and more positive. So let's add this to our graph. So the membrane potential is going to increase because all of these positive ions are entering the cell. Well, just based off of kind of the English language, you can probably figure out what the name of this step is. All right, if this is called polarized or polarization down here, as more sodium comes into the cell, this is called, I'm just going to go ahead and type this, this is called depolarization, right? As sodium comes in, as sodium comes into the cell, right? What happens next, because this message is propagating down the axon, what you're also going to notice is that, well, hey, you've got these potassium voltage channels too, right? It's not just the sodium, you also have potassium, but they are much slower, right? They are much slower than the sodium channels. So this one I'll go ahead and do in red, right? So once the wave has passed, once this electrical gradient, this impulse has passed, the potassium channels are like, oh, I guess it's our turn to open. We're so slow. So after this signal has passed, then these potassium channels open. And again, in real life, this happens, boom, very quick. But for us, we're going to talk about it like it's taken a hot minute here. Oops, I lost my. I hate this. This is frustrating. I lost my little marker here. So as this change in voltage passes these potassium channels, they open a little bit late. They're a little bit late to open. But once they do open, the high concentration of potassium inside the cell diffuses to a lower concentration of potassium outside the cell, right? So now positive ions, in this case potassium, are leaving the axon, right? They're leaving the axon. And as this signal propagates down the axon, more and more of these potassium voltage-gated channels open, right? So because this is a slower process, these channels are a little bit slower than the sodium channels, what happens, go back to our graph, and I'm just going to keep it in red, but you don't have to change color in your graph, is that the voltage inside the cell drops. And because the potassium channels are so slow in comparison to the sodium channels, it actually drops below negative 70 millivolts. It actually drops below negative 70 millivolts. So this little kind of drop here, where you go below the baseline, is called, get this, it's called hyperpolarization. Right? This section right in here is called hyperpolarization. And the process of the voltage potential dropping back down as potassium leaves the axon is called repolarization. as potassium goes out. One thing I did forget to mention is that as sodium leaves, the channels do close back up, and as potassium as sodium enters and as potassium leaves, those channels also close. So at the top here, you can go ahead and add that. Sodium channels close, and potassium channels open. Right. And then eventually they return to their baseline. Once the potassium channels catch up to the sodium channels, it returns back to the baseline negative 70 millivolts. Right. So that's how this electrical signal travels down the axon. That's how it travels down the axon. So what you're going to notice is that there are two things happening here. There are two things happening. You have the electrical gradient, the electrical signal, and that's because of the difference in charges and how that charge changes as these channels open and close. But you have the electrical gradient because of the chemical gradient, because of the different amounts of, in this case, sodium and potassium moving in and out of the cell. Right? So both of these electrical and chemical gradients are in charge of sending this electrical impulse down the axon. That impulse is called the action potential. Right? The whole change in voltage moving down the axon because you've changed the chemical gradient is called the action potential. So the change in the membrane voltage down the axon, that sends the signal, right? It's called the action potential. So think of it as a wave, right? Like doing the wave in a crowded stadium. You send that signal propagating down the axon. All right, well, eventually that signal reaches, I'll go back to blue here. All right, potassium and blue. Yeah, I'll go back to blue. Eventually that signal reaches these calcium voltage gated channels. Calcium voltage gated channels. Well, calcium is in a higher concentration outside the cell. So when the voltage hits these channels, calcium is going to diffuse in. Calcium is going to diffuse in to the end of this cell. And what's going to happen is it is going to cause the exocytosis of these vesicles. Right? These are vesicles that contain what are called neurotransmitters. These are proteins that are going to signal kind of the adjacent cell. So here are the steps to that. And let's add the steps up here. So the first step, step 1, is that calcium, as it diffuses into the original host cell, the original cell on the left here, calcium channels open and calcium moves into the cell, the neuron. The second step, so that's step 1, right? That's right here, step 1. Step 2 is that calcium causes the exocytosis of these neurotransmitters, causes the exocytosis of these neurotransmitters. And these neurotransmitters move along cytoskeleton. Right, so remember that they're not just kind of floating around here. There is cytoskeleton that these vesicles move along, and they fuse. So they fuse with the membrane of the cell, and they release those neurotransmitters into the synapse. Right? They release the neurotransmitters into the synapse, the space between adjacent neurons, right? And this is important. This is just diffusion. This is just diffusion. There's a high concentration of neurotransmitter coming out of the vesicle, and it diffuses into the synapse. right? So that is step 3. The neurotransmitter, I'm going to abbreviate that, is released. So that was step 3. And then step 4 is that the neurotransmitter diffuses across the synapse. All right. Well, this is another neuron. It's a neuron just like the neuron we saw before. So let's go ahead and remember that the inside of the neurons are negatively charged. All right, I'm just labeling kind of what the other neuron had in the big sheet. And the outside, the extracellular matrix side, is positively charged. Go ahead and add these to your big sheet. This is called the postsynaptic neuron. It's the neuron after the synapse. But again, it's the body of the neuron, so we're out where the dendrites are. Here's the nucleus with the DNA in it. And here are ligand, if you guessed it, ligand-gated ion channels. Ligand-gated ion channels. And if this looks familiar, it's because these are the exact same ligand-gated ion channels that we started with. And here's where the ligand comes from. The neurotransmitter that was released from the presynaptic portion of the membrane when the vesicle fused and then diffused throughout the synapse, that's the ligand that then binds to the ligand-gated ion channel in the adjacent neuron, right? So this ligand that we started with is a neurotransmitter from the previous neuron. That's where it comes from, right? And exactly what you think is going to happen, right? So when that ligand binds to this ligand-gated ion channel, remember this is a sodium channel, sodium, I'll just draw it here just to, I'm a little bit crowded up here. Sodium is going to rush in. Remember, salty banana, so salty outside, not salty inside. Sodium rushes in down its concentration gradient, diffuses into this cell, and starts to turn the inside of this next cell positive. And you're right back to where you started. Then you propagate another action potential down the adjacent neuron, and it's the whole process all over again in the next neuron, right? That's how this signal is sent from cell to cell, from neuron to neuron, right? So in this case, calcium, the role of calcium is a second messenger. Remember, we talked about this last video. Calcium is a second messenger in this system. All right, so we're almost done here. You might have realized that we're not quite done because as is, oops, why does it do that? As is, we have, I guess, a potassium-y salty banana thingy kind of going on, right? All of the potassium is now outside the cell, and all of the sodium is now inside the cell. So we have to switch these back and restore the cell's default concentrations of sodium and potassium. And the way we do that, you guessed it, is right here with these protein pumps, these sodium-potassium pumps that are all over the axon, right? So at the very end, all of these pumps are going to restore the original concentration of potassium and sodium inside the cell. So a lot of potassium back inside, that gets pumped back inside, and all of the sodium gets pumped back outside, and it restores the original baseline concentrations, right? And again, this happens, boom, very quick, the entire thing. Starting with the ligand, all of these changes in chemical gradients and the voltage changes all the way down through the axon, the synapse sending off neurotransmitters, and the resetting of the charge using the pumps, the sodium potassium pumps, that entire process takes split second, fractions of a second, right? And the cell is ready to send another message again. And again, this is a process that once that chemical gradient and that electrical gradient gets started, you cannot stop this. You cannot control the electrical impulse. Drugs that we take that modify neurotransmitters affect the synapse. So this is something, the synapse is actually where we have the ability to regulate this process. And this is where like a lot of prescription medications that affect the brain, like SSRIs and stuff, affects neurotransmitters in the synapse, right? But the actual impulse itself, once that impulse starts, you cannot stop it. It goes to completion, and then the cell resets quite quickly. So we're going to stop there for right now. I hope this helped, and I would get to the point where you can confidently explain what's going on, right? And that's just going to take some practice, and I'll see you next time.